Hello, and welcome to the season premiere of the Molecular Chat with Nika and Mike. I'm Nika.
And I'm Mike, and the goal of our show is to help scientists, just like you, understand molecular mechanisms by visualizing them.
Today in the Molecular Chat, we'll be looking at the NFAT pathway induced by T helper cells and its effects on the immune system.
But before we dive into the molecular world, let's briefly recap the history of how this pathway was discovered. Studies in the 1960s and 70s demonstrated that B and T lymphocytes were primarily responsible for antibody production and cell-mediated immune responses.
They were discovered using animal models, clinical evaluations of patients with immune disease, and new technologies such as immunoblotting and X-ray crystallography.
Later in 1988, the NFAT transcriptional pathway was discovered in T cells by J.P. Shaw et al. In 1992, Clipstone and Crabtree found that NFAT transcription factor activity was upregulated with increased levels of calcineurin.
Over the next few decades, the rest of the signaling cascade was discovered, starting with antigen binding to the T cell receptor and ending with NFAT translocation into the nucleus to activate the T cell.
So now that we all know a little more about the topic, let's dive right into the molecular world.
Let's start with a closer look at what a T helper cell looks like. First is the phospholipid bilayer, which also consists of lipid raft regions important for signal transduction. In order to activate the T helper cell and transduce a signal, we need a T cell receptor, TCR, and a CD3 complex, which consists of three co-receptors with immunoreceptor tyrosine activation motifs, ITAMs. ITAMs are regions on the receptors that can be phosphorylated to amplify a strong enough signal to be transduced. Another key player is a CD4 co-receptor, which is T helper cell specific. Although we will be using a slightly simplified model today, remember that this binding also requires other co-receptors. Keep in mind, lipid rafts are rich in tyrosine kinases such as LCK and phospholipids such as phosphoinositol bisphosphate PIP2.
Here we have an antigen-presenting cell with an MHC class II molecule presenting a viral peptide on its surface. The TCR on the T helper cell must be specific for the virus in order to activate the T cell. Once the TCR is presented with the antigen, all necessary players move to the lipid raft region. So keep in mind from this scene on, we are in a lipid raft. Next, we have the CD4 receptor bind to the MHC class II molecule, which offers a second binding interaction that helps hold the entire complex together for longer to increase downstream signaling.
The LCK tyrosine kinases and lipid raft phosphorylate ITAMs on CD3 co-receptors, leading to adaptive protein recruitment and a phosphorylation cascade. The first adaptive protein which is recruited is FIN. FIN gets phosphorylated and in turn recruits ZAP70, which is then phosphorylated and continues the phosphorylation cascade.
Here we have our T helper cell again, with ZAP70 now recruited and activated. ZAP70 phosphorylates a membrane-bound adapter protein, LAT, which binds adapter protein GAD constitutively associated with adapter protein SLP76. This complex goes on to phosphorylate phospholipase C-gamma. PLC is an enzyme necessary for phospholipid cleavage.
PLC goes on to cleave PIP2 into two products, inositol triphosphate, IP3, which is now free-floating in the cytoplasm, and diacylglycerol, DAG, which remains membrane-bound. This entire process requires multiple TCRs to be activated and multiple IP3s to be produced in order to activate the NFAT pathway.
Here we have the IP3 molecules that are in the cytoplasm moving towards the endoplasmic reticulum. Once they bind to their specific receptor on calcium channels, the IP3 molecules signal calcium channels to open, releasing calcium ions into the cytoplasm.
Calcium ions released by the ER then bind to the calmodulin protein, which changes from inactive to active conformation. The calcium calmodulin complex binds and activates calcineurin, a protein phosphatase. Active calcineurin can now cleave a phosphate off of NFAT. Without this dephosphorylation, the NFAT transcription factor would not be able to translocate to the nucleus.
Once NFAT is dephosphorylated, it translocates into the nucleus and can finally do its job as a transcription factor. So to recap, NFAT activation begins with antigen presentation and ends with NFAT binding to DNA to activate genes that produce a variety of cytokines such as IL-2, IL-4, and IFN-gamma. These cytokines play an important role in producing an adaptive immune response to fight off the invading antigen. Specifically, These cytokines help other T and B cells to proliferate and differentiate.
Welcome back! We hope you've enjoyed the molecular world of the NFAT pathway. Before we part, we wanted to mention a couple ways this pathway is important in development and treatment of immune diseases.
First off, the NFAT activation pathway amplifies adaptive immune responses. Unfortunately, this can backfire if T helper cells are overstimulated or misdirected to attack self, leading to tissue damage.
This is commonly seen in autoimmune diseases such as rheumatoid arthritis, multiple sclerosis, and type 1 diabetes. It's also common in organ rejection after transplantation. Fortunately, the NFAT pathway can be inhibited by immunosuppressant drugs to treat some of these diseases. For example, cyclosporin A and tacrolimus are used to treat rheumatoid arthritis and can prevent organ rejection after transplantation by inhibiting calcineurin.
Studying the molecular mechanics of the immune system is the gateway to understanding how immune diseases develop, how to prevent them, and how to provide safe and efficient treatment.
Well, it sounds like that's a wrap. Thanks so much for watching Molecular Chat with Nika and Mike.
Until next time.
Till next time!